100 years ago, improved microscopes opened a new world to investigation, the internal world of the cell. Seen for the first time within cells were tiny bodies called organelles. But was there more to be seen? The problem was that smaller structures would also be smaller than the shortest wavelengths of light, and so could not be seen. This limit, imposed by light, literally left biologists in the dark when it came to observing the inner workings of cells, particularly in the region of the cell called the cytoplasm. Cytoplasm is that part of a cell within the plasma membrane, but not including its nucleus. This is the cytoplasm. Cytoplasm is made up of a watery fluid, the cytosol, containing the cell's supply of dissolved material. Suspended in the cytosol are the cytoplasmic organelles. The internal structure of even large organelles remained a mystery until an imaging system was developed that used an illuminating source with much shorter wavelengths than light. The electron microscope gave biologists their first detailed look at the inner structure of cytoplasmic organelles. Discovering the function of these bodies continues to be a major goal of cell biologists. Their investigations often begin by first breaking up cells. They can then isolate batches of a particular kind of organelle by taking advantage of small differences in density between the various cell parts. Once isolated, an organelle's function can be explored using a variety of techniques. This approach showed that major life functions like cellular respiration, digestion, and protein synthesis are each associated with a particular kind of cytoplasmic organelle. Many of the processes going on in the organelles of a cell involve the formation or breakdown of macromolecules, giant molecules made from smaller molecular building blocks. For example, simple sugars are the building blocks that make up polysaccharides. Fatty acids are the building blocks of fats. Amino acids are the building blocks of proteins. Nucleotides are the building blocks that bond together to form the nucleic acids, RNA and DNA. The cells of animals differ from plant cells in one important way: their method of acquiring their building block molecules. Animals pick up most of their building blocks from the original manufacturers, plants. In an animal's intestine, the plant's macromolecules are broken down into their building blocks during digestion. These smaller molecules are absorbed into the animal's bloodstream and carried to its cells. However, simple animals often carry out a more direct kind of digestion within individual cells. When this primitive animal, Hydra, swallows its prey, the prey undergoes preliminary breakdown in Hydra's sac-like digestive cavity, releasing bits and pieces of food that are then engulfed and digested by individual cells lining the gut. This kind of digestion within cells, is best seen in organisms that are made of but a single cell. This one, Vorticella, gulps in bacteria which are then broken down to their building blocks entirely within the cell's cytoplasm. During this process, the bacteria are encased in a pocket formed by a piece of plasma membrane that pinches off and circulates around the cell. These food-filled membrane bubbles, or food vacuoles, contain digestive enzymes. The enzymes actively break down the bacteria's macromolecules into their building blocks. Special dyes can provide a clue to the conditions necessary for this breakdown to occur. This indicator dye turns deep red in acid solutions and amber in neutral solutions. When the dye is applied to living cells, it concentrates in food vacuoles. The deep red color of newly formed vacuoles shows that they're acidic, important because digestive enzymes work most efficiently under acid conditions. The digestive enzymes come from small, dense particles that occur in the cytoplasm. These organelles, called lysosomes, are particularly abundant in cells that ingest their meals. A lysosome is basically a sack of powerful digestive enzymes that can be dumped into a food vacuole. Its membranes also contain protein pumps that pump hydrogen ions into the vacuole, making them acidic and so assuring effective digestion. Digestive enzymes are only one of many kinds of proteins manufactured in the cytoplasm. These enzymes, like other proteins, are assembled on tiny organelles called ribosomes that dot the membranes surrounding the nucleus. This region of the cytoplasm is called the endoplasmic reticulum. It is the cell's major site of protein assembly. Once manufactured, digestive enzymes travel to the Golgi apparatus, where they are packaged in sacs of membrane and enter the cytosol as lysosomes. Lysosomes are key players in our body's defense system. Here, a human white blood cell pursues a harmful bacterium. Eventually, the bacterium will be engulfed and broken down into its component molecules by a swarm of lysosomes within the white blood cell. Lysosomes also help to recycle a cell's worn or damaged parts. As a worn-out organelle is digested in the lysosome, its building block molecules are returned to the cytosol. Many of these building blocks, whether acquired from the digestion of food molecules or by recycling of worn-out parts, are used to supply the cell with energy. Extracting this energy from building blocks takes place in mitochondria, structures that occur by the hundreds and thousands in the cells of plants, animals, and protists. A mitochondrion is composed of two layers of membrane. The inner membrane is folded, extending into the organelle's central cavity, increasing surface area for chemical reactions. In 3 dimensions, the organelle looks like this. The cutaway shows the 2 layers. In living cells, mitochondria can be seen concentrating in areas where energy needs are greatest. In Vorticella, they line up along the spring-like stalk, ready to power the contraction that may snap the cell out of danger. In muscle cells, the mitochondria lie close to the contractile fibers. Plant cells are loaded with mitochondria that extract energy from sugars and fatty acids made by photosynthesis. To release the energy contained in food molecules, a mitochondrion must also take in oxygen. In this process, called aerobic respiration, the key energy events occur on the knobby lining of the inner membrane. Here, the energy released from sugars and fatty acids is used in the formation of ATP, the universal energy carrier. An ATP molecule holds this energy in a powerful chemical bond. The reaction is reversible, so that the ATP can donate energy to drive reactions anywhere in the cell, and then be recharged at another mitochondrion. While mitochondria are found in all types of nucleated cells, chloroplasts, life's primary energy transformers, are only found in plants and green protists. Within a chloroplast, the electron microscope shows stacks of inner membranes organized like arrays of solar cells. Layers of chlorophyll molecules capture the light that drives photosynthesis. In a plant, much of the energy gained through photosynthesis is used to split water into hydrogen and oxygen. The hydrogen is used in a complex chain of reactions that produce carbohydrates and fats. The oxygen is given off as a waste gas, adding oxygen to our atmospheric reservoir. While carbohydrates and fats provide all cells with energy, proteins are the macromolecules that form much of a cell's structure. And as we saw, proteins called enzymes control the thousands of chemical reactions that go on each second in the cytoplasm of every living cell. We have also seen that proteins are assembled from amino acid building blocks. The amino acid sequence for each protein is stored on string-like molecules of DNA in the nucleus. Along the DNA molecule, each amino acid is represented by a code word or codon consisting of 3 nucleotides. The code is copied onto a closely related nucleic acid, RNA. This messenger RNA migrates out of pores in the membrane surrounding the nucleus, moving toward the endoplasmic reticulum, where it connects with one of the many ribosomes. Here, transfer RNA, also from the nucleus, identifies individual amino acids which are assembled in strict accordance with the sequence of codons carried on the messenger RNA instruction tape. The chain grows, creating the protein macromolecule that will eventually play its role in cell chemistry. The completed proteins move to the Golgi apparatus, where some are packaged into lysosomes and others released directly into the cytosol. Some, like hormonal proteins, are exported into the bloodstream, providing communication between cells in different parts of the body. So, in its overall function, the cytoplasm resembles a factory. It breaks down complex materials into their building block molecules, which can be reassembled into new macromolecules or used for energy. And while we know the basic outline of how the cytoplasm carries out these processes, there is still much to be learned about its control systems and how it segregates and transports the many different kinds of molecules essential for the life of a cell.